Okay, thank you so much to everyone for being here after this heavy lunch. Today I'm going to be talking about an unusual tool to analyze biological information, but I hope I can get the message to you. First, I'd like to bring your attention to a phenomenon that has been reaching awareness lately in the plant pathology community, and that is the ability of viruses to jump across kingdoms of life. And I'm not talking just about a virus that jumps from animals to humans, that we know many examples of that, but between different kingdoms of life, that is from fungi to plants and even to insects. Here you have an example to the left, one of the first experimental assessments of this phenomenon, in which a fungal virus was able to infect a plant, systemically spread inside the plant, and then transfer to a different fungal species, and even transfer to other plants, and even transfer to insects. And this was an actual experiment. To the right, you have some examples of these viral families that have this ability. And truth is that roughly 80% of biomass on Earth are plants. So plant viruses probably have a large impact on ecosystem stability and are not just these agents of destruction that we are used to think about it. We can combine this with this globalization phenomenon that is provoking disruption in our forests. And disrupting forests also disrupts the viral flows in the ecosystem. Here you have an example of cross-kingdom virus outbreaks, many between animals, but also from pathogenic fungi to plants. So this is a phenomenon that we are worried about in plant pathology community, which is, are there any possibilities that the next COVID, for example, might come from forests, taking into consideration that we are heavily disrupting forests? So, one way to assess this issue was, okay, we can do this experimentally, we can go to the field and collect data, but probably this would be a huge amount of work and we have no clue so far. So why not do a first approach in silico? And we got this idea from this paper you have there, which uncovered unexpected roles of blood glucose levels in the severity of symptoms of COVID. And they achieved this not by analyzing the data, not by analyzing the results. They did this by analyzing the manuscripts, the texts themselves. So we were thinking, okay, maybe in the literature of plant pathology there are already some hints that are hidden that we are not aware about, but likely there are machine learning tools that nowadays can understand texts and can uncover relationships within the text itself, not in the data. This branch of machine learning is called natural language processing. Some of you might have heard of it. Lately, ChatGPT-4 and this kind of chatbots and conversational large language models are ubiquitous. They can process posts in social media, they can process your emails, we use spam filters. Nowadays, this branch of machine learning is huge and has great advantages. So, would it be able to, instead of using structured data, which are data sets, something that is stored in a database, that is referenced, that is in tabular format, that we are used to that, could we extract knowledge from unstructured data to aid in this search for a candidate, for a viral candidate for the next COVID from the forest, likely? That was our objectives. Can artificial intelligence uncover biological associations using raw human readable text instead of the datasets? We followed 3 steps. The first one is your typical collection of papers, as you would do in any systematic review. You have your keyword collection. And then, because we have so many, 3,500 abstracts which were allowed, we said, okay, maybe we can do some filtering, as you would do in your normal systematic review, but instead of doing it by a human, we use an automatic made bias classificator, which is really easy to train. This classifier decided for us which abstracts were relevant or not for our question, and we ended up with almost 2,000 abstracts, which was quite some huge amount of data yet. The next step was the natural language processing in itself, and the most important module in this natural language process is the name entity recognition model. Those are machine learning models that are pre-trained in biomedical nomenclature, in biological taxons, in biological entities, because human language is noisy, and sometimes the same biological entity might be named by the binomial species, sometimes by the vernacular name, sometimes by a common name, sometimes you abbreviate things. So researchers, when they write the papers, they are not using all the time the same name to refer to the same biological entity. So that's why we need machine learning models, these named entity recognition models that can generalize and can catch these small details instead of using just a dictionary of terms. And with these named entity recognition modules, we end up with a list of biological entities, as you can see here in the highlighting paragraph. Protoplasm is a cell component, virions is an organism, Ursuline is an organism, fungi is a taxon, hosts are an anatomical system, so it depends on the categories you train the model of, you end up extracting those categories from your text. Then we have sentences. In each sentence we have the biological entities we found, and my favorite part was actually this last step in the methodology, which has to do with assigning a positive or negative sentiment to the sentences. This is really similar to what Amazon does to the product reviews, whether this review is positive or negative. This is the same, and this is how we wanted to actually uncover this relationship. Are these viruses appearing more frequently associated to this plant in negative sentences or in positive sentences? And maybe we found out something that we were not aware about. So, as a fair result, we got this comparison against the Fick law. Fick law is the expected frequency distribution of natural language, where you have a really common word, and then you have this drop down. So, if your text is enriched in terms that belongs to what you were interested in, you will deviate from the Fick law, and this is like a good measure of your approach was robust. At least your text is enriched in something different from your natural language. These were biological terms: virus, plant, vector, protein, viral, infection, whatever. So first robust step, okay. Then we have the two named entity recognition models, and because each model had different categories, we had to somehow find the common place for organisms, which was organism and taxon, and we selected only 6,000 entities that were classified simultaneously as organism and as taxon. So those look like the thing we were looking for, which were viruses, plants, fungi, insects, and so on. And this kind of network is what we end up with. One network for positive sentences and another network for negative sentences. There are many commonalities, which were kind of expected because, you know, this was kind of a proof of concept, but there are also different things between them. For example, in positive sentences, the relationship between plant and animals is direct, but in negative sentences, the relationship between plant and animal is mediated by rhabdoviruses and whiteflies. So there is, take into account that this comes from pure text, from abstracts. This is not data, this is not the results. So this natural language processing pipeline was able to somehow capture that animal and plants might have a negative relationship if the rhabdovirus is involved in that relationship. So we're still in the phase of analyzing what is going on. This is, as I told you, a proof of concept, but I think there is great potential in these tools to extract early concept, early candidates, and then you go for experiment, and then you go to the field and do your sampling and whatever, but as a first step, I think it's a great tool. For sure, I don't know you had the time to take a look, but many of the viruses we found out were agricultural viruses instead of forest. For sure, this needs improvement. We have to put the focus on the forest to try, to extract information more similar to what we were interested in, but as a first approach, I think it's quite good. And I don't know if I have a couple of minutes left. Okay, because I wanted to say some words, particularly to the master and PhD students. This project to me was like a side project. I did this in my spare time. I just wanted to try out different algorithms, types of data that I never used before. And I think it's a good idea for computationalists to, from time to time, allocate some moment to side projects that has nothing to do with your real project, your real focus, to try to expand your abilities. And honestly, in the past months, this project was one of the moments where I had the most genuinely fun analyzing data. And it wasn't my main occupation at the moment, but it was fun because it was something I decided to do as a side project, and I think it's a good habit to have. And I think that's all. Thank you so much for your attention.